In this G0 phase, the cells try to remain inactive and then they go back to the S phase, and that G0 phase is called as the Quizen stage, wherein the cells are quite inactive. In zygote, what happens is that those condensed chromosomes, they tend to start pairing, and that is called as synapsis. In anaphase, what happens? The sister chromatids are pulled apart from the metaphase plate and they move towards the opposite poles of the cell. Hello everyone, a warm welcome to this session on revision class for first PUC Biology. I'm Dr. Divya, Biology faculty, Vidhyaashram Pre-University College, Temple of Excellence, Mysuru. So in this session, we'll learn about the chapter that is cell cycle and cell division. So here we'll study about what all are the contents that are present in this particular chapter. We'll have a brief discussion regarding that is under synopsis, and we'll learn the important 3-mark question and answers that will appear from this particular chapter. So one 3-mark question will be asked from this particular chapter, and also apart from the 3-mark question and answer, multiple choice question wherein 3 multiple choice questions of 1 mark each will come from this particular chapter. So therefore, this chapter carries a weightage of 6 marks. So in this session, we'll concentrate on the synopsis and the 3-mark question and answer. In the session 2 of the continuation of this revision class, we will concentrate on the multiple choice questions. So talking about the cell cycle. So in this chapter, we need to study about the cell cycle, wherein we need to study about the mitotic stages and the meiotic stages. So when talking about the cell cycle, in the cell cycle, there are 2 important phases that occur. One is the interphase and the other one is the M phase. So in interphase, it is divided into G1 phase, S phase and G2 phase. So before the cells entering into the mitotic cell division or meiotic cell division, they have to finish a cycle, cell cycle called as interphase, wherein in the interphase 3 important stages are there. That is the G1 phase, which is also called as the gap 1 phase, the S phase, which is also called as the synthesis phase. It's called the synthesis phase because it is in the S phase that the DNA synthesis or the DNA replication starts. So that is why. And the last one is the G2 phase, that is the gap 2 phase. Now some of the cells from the G1 phase, instead of entering into the S phase, they go into a phase which is called as G0 phase. In this G0 phase, the cells try to remain inactive and then they go back to the S phase, and that G0 phase is called as the Quizen stage, wherein the cells are quite inactive. Next, moving to M phase. Now, after completing the interphase, the 3 phases under the interphase or 3 stages of cell cycle under the interphase, the cell, it moves into the M phase. So M phase is nothing but mitotic phase, which is also called as equational division. Why equational? Because if the parent cell has, say for example, 20 chromosomes, then the daughter cells will also carry 20 chromosomes. So equal number. So 20 times DNA replication will occur, wherein 20 DNA strands will get produced, which will move into the daughter cell. So therefore, parents will also have 20, daughter will also have 20. This is just an example that I'm giving. So therefore, mitotic phase means it is equational division. Next, in mitotic phase, there are 2 important stages that occur. One is karyokinesis and one is cytokinesis. So karyokinesis meaning the nucleus. So division of the nucleus or the nuclear content, we call it as karyokinesis, and division of the cytoplasmic content, we call it as the cytokinesis. Now in karyokinesis, there are different phases. One is the prophase, the second one is the metaphase, then comes the anaphase and the telophase. Now, so in prophase, what happens is that the condensation of the chromosomes. So usually the chromosomes, before entering into the mitotic phase or the prophase, they'll usually be thread-like and haphazard distribution will be there. Those thread-like chromosomes now start thickening and condensing in size. After the condensation of chromosome, whatever centrosome is there, that will start moving towards the opposite poles of the cell, and after the movement of the centrosome to the opposite poles of the cell, asters will get formed at the opposite pole. And the formation of the asters is quite important. Why? Because it is those asters that give out the spindle fibers which are needed for the traveling or proper distribution of the chromosomes to the daughter cells in the next stages of the mitotic cell division. After completing prophase, the cell next enters into the metaphase. Now the metaphase, we got to know that in the prophase what happens, the condensation of the chromosome will occur. Now those condensed chromosome need to get aligned properly in a single plane so that in the next stage it can distribute to its respective places. So what happens here is in the metaphase, the spindle fibers will be formed and it will get attached to kinetochores. So kinetochores are nothing but small disc-like structures which are made up of proteins that are present in the center of the chromatid arm or the chromosomal strands. Now, after the formation of the spindle fibers, the chromosomes that are there, which chromosomes? Those thickened chromosomes which are there in prophase, right? They will come and get aligned at one plane in the center, or which is called as, they get aligned along the metaphase plate. That is what happens in the metaphase. Now in the metaphase, what is that? Chromosomes and all have aligned in the metaphase plate. Then the asters have produced the spindle fibers. The spindle fibers are attached to the kinetochores present in the chromosome. So much has occurred. After this, it will then enter into the anaphase. In the anaphase, what happens? The centromere. So what is centromere? Centromere are nothing but present along with disc-shaped structures called kinetochores. And the main function of the kinetochore is to attach itself to the spindle fiber. The function of the centromere is to hold the 4 chromatid arms that are there. So in chromosomes, we know that it has chromatid arms like this. Now the chromatid arms are held by structures called as centromere. So these centromere, what happens? It splits. So when it splits, what happens? The chromatids or the chromosomal arms, they tend to split and they will start moving towards the opposite pole. So that is what happens in the anaphase. Now after the anaphase, it will then enter into the, now in the anaphase what happens? The chromosomes which were aligned in the metaphasic plate, they'll start to split and they'll start moving towards the opposite pole. That is what happens. Now it enters into the telophase. Now the chromosomes and all that, they have finished moving properly towards the asters or towards the centrosome or towards the opposite poles, and they form clusters or groups there. And after that, what happens? Nuclear envelope will start to develop because it has to have its own nuclear envelope, no? So nuclear envelope will start to develop. Nucleolus will get formed. Golgi complex will get formed. Endoplasmic reticulum, all that will get formed there. So therefore, telophase begins or it is nothing but a marking that the cell has divided. So that is what happens in the telophase. After completing telophase of mitotic cell division, let's study what happens in meiosis. Meiosis is nothing but called as reduction division, because in meiosis what happens? Always the daughter cells will not have the same number of chromosomes as that of the parent cell, what we had seen in mitosis. Here in meiosis, the daughter cells will always have half a set of chromosomes. Say for example, if the parent cell has 20 chromosomes, then the daughter cells each will have 5. That is half a set. So that is called as meiosis. Therefore, it is called as reduction division. The chromosome number tends to get reduced there. So in meiosis, again, there is meiosis 1 and meiosis 2. First, it will complete the entire steps under meiosis 1. Then it will enter into interkinesis. And then it will enter into meiosis 2, and those cells which have completed the stages under meiosis 2 will enter into cytokinesis. Now talking about meiosis 1, in meiosis 1 also there is prophase 1, metaphase 1, anaphase 1 and telophase 1. So all the steps are same like that of the mitotic cell division itself. So in prophase 1, again, they undergo different stages. One is leptotene, then comes zygotene, then comes pachytene and diplotene, and the last one diakinesis. So in leptotene, what happens is that the compaction or the condensation of the chromosome occurs in leptotene. In zygotene, what happens is that those condensed chromosomes, they tend to start pairing, and that is called a synopsis, wherein a synaptonemal complex will get formed, which is a railway bridge-like structure, which is again made up of protein, and therefore bivalent tetrad chromosomes will get formed, wherein the chromosomes will have 4 chromatids or 4 chromosomal arms. After that, tetrad formation will occur in pachytene and recombination nodules will get formed there. So recombination nodules need to get formed so that where exactly the crossing over should occur between the non-sister chromatids, that place exactly is marked by the recombination nodule. So wherever recombination nodules are there, they're only crossing over between the non-sister chromatid chromosomal arms also I can say, or non-sister chromatids will occur there. Therefore, here crossing over will occur, wherein in the recombination nodules, recombinase enzyme will get produced. Always remember, whenever anything ends with ASE, in biology, it means it is an enzyme, okay? Because always the name of an enzyme ends with ASE. So, recombinase is an enzyme which is produced in the recombination nodules that will help in the crossing over in that particular recombination nodule place. Now, after that, after the crossing over, it will enter into diplotin, wherein in diplotin, synaptonemal complex that was formed, that is, I told you, railway bridge like structure that was formed, it will get dissolved. It will just get removed, and in that place, chiasmata formation. Chiasmata formation means the chromosome will start looking like a X, because crossing over occurred between the non-sister chromatids were in the packetine. So that is why. After diplotin, then it moves to diakinesis. So diakinesis is nothing but the chiasmata formation will stop and the nucleolus will disappear. Nuclear envelope will break. After that, it will enter into interkinesis. After interkinesis, it will enter into meiosis 2. Again in meiosis 2, what happens? Prophase 2, metaphase 2, anaphase 2, telophase 2 will occur, and later on cytokinesis will take place. Same like the mitotic cell division. So these are the different processes that occurs in a cell cycle. So now we have an idea of what all are the contents present in the chapter. We'll go on to studying some important 3 marks question and answers. So the first question, distinguish between prophase and telophase. Now when they say prophase and telophase, you can write the general characteristic that are there under that. So prophase, first what happens? Condensation of chromosome. So if you forget the word condensation, you can also write thickening of the chromosomes or shortening of the chromosomes will occur in prophase stage, and the nuclear membrane will start to disappear. Of course, in the prophase, the nuclear membrane should start to disappear, because whatever nuclear content, especially the chromosome is there in the nucleus, it has to come out of the nucleus, right, for division purpose. So that is why nuclear membrane starts to disappear. Then nucleolus will start to disappear, and here the chromosomes are present at the opposite poles of the cells. Or chromosomes, to be specific, we can say that the centrosomes. Centrosomes are present at the opposite poles of the cell. So moving to here, it's the differences. So talking about telophase, there in prophase, condensation of chromosome will start. Here, the condensed chromosomes will start to decondense. Next in prophase, the nuclear membrane will start to disappear, but in telophase, the nuclear membrane will start to form. Nucleolus starts to disappear in the case of prophase. In here, the nucleolus appears, that is in the daughter nuclear membrane, or the nucleolus also will start to appear. Their centrosomes are present at opposite poles of the cell, or here we can say chromosomes are randomly arranged in the cells in the case of telophase. So next question, explain interphase with its stages. So interphase is the process of cell cycle, that is before entering into M phase or mitotic phase. There are 3 stages, which is there, that is the interphase. So there, interphase is the process of cell cycle when a cell prepares itself to divide or to duplicate, and most of the life of a cell is present in the interphase, because first it gets ready in the interphase and then only it enters into the M phase. So there are 3 phases involved in it: G1 phase, S phase and G2 phase. So G1 phase, it takes place right after the division of the cells, and a lot of protein synthesis will occur during the G1 phase. In S phase, what happens? It suggests that it is a synthesis phase. Therefore, DNA replication begins in the S phase. This is one of the very important MCQ question. Next, G2 phase, the cell undertakes preparation to enter into mitosis. So once G2 phase is complete, then the cell enters into the mitotic phase of cell division. So third question, with a neat label diagram, compare metaphase and anaphase of mitosis. So you have to draw the diagram of metaphase and anaphase. It's quite easy. You need to draw the centrioles, then these are the asters, then you can draw the spindle fibers, and this is the centromere, then the chromosome, all this, and you have to draw the chromosome on a plane, that is a metaphase plate, it will be aligned. And for anaphase, the same thing, but the chromosomes would have moved to the opposite pole. Write a note on both of these. So metaphase, the microtubules of the spindle fibers have attached and the chromosomes have lined up in the metaphase plate. Or if you just write the chromosomes line up in the metaphase plate also, it is fine. In anaphase, what happens? The sister chromatids are pulled apart from the metaphase plate and they move towards the opposite poles of the cell. So much you need to write. So next question. Distinguish between zygotine and pachytene of prophase 1. Of what prophase 1? Because in prophase 1, we had studied the different stages. So we had the leptotene, the zygotine, the pachytene, then diplotin and the diakinesis. So much. So here we have to distinguish between zygotine and pachytene. Same thing they can ask for leptotine and zygotine, leptotine and pachytene, like that also questions can be asked. So here, because the question is distinguish between, we need to make 2 columns. On one end you write zygotine and on the other pachytene. Now let's write the differences. In zygotine, homologous chromosomes which are there, they begin to pair. So, meaning the chromosomes which carry the same genetic material, they tend to pair. Next here, 2 chromatids of each pair tends to separate here in the case of pachytene. In zygotine, synapsis begins in, synapsis formation occurs in the homologous chromosomes. Here, crossing over occurs between non-sister chromatids. In the case of zygotine, chromatin condensation will not occur. In the case of pachytene, chromatin condensation begins. So these are the differences. 3 differences each you need to write. Moving further, name the stage of cell cycle at which one of the following event occurs. So they have given a number of questions here. Like this also questions can be asked, wherein chromosomes are moved to spindle equator. Where does it occur? Chromosome movement to spindle equator. So whenever the word come equator means at this, we can say metaphase, because chromosomes move towards the metaphasic plate. Next, centromere splits and chromatids separate. It occurs in the anaphase. Next, pairing between homologous chromosomes will start in zygotine of meiosis 1. Next, crossing over between homologous chromosomes will take place in pachytene of meiosis 1. So, moving further, explain synaptonemal complex and metaphase plate. So, synaptonemal complex, these are zipped structures, or you can write railway bridge-like structures that are formed between 2 homologous chromosomes during the prophase of first meiosis, or you can write during the prophase 1 of meiosis, and the continuous arrangement of the chromatin throughout the meiotic prophase, such as recombination, condensation and homologous chromosome malfunction are linked to this disassembly and assembly. So that is nothing but synaptonemal complex, wherein all the homologous chromosomes are held together by the synaptonemal complex, which is a bridge-like structure. Next, talking about the metaphase plate. In metaphase, the centromeres of the chromosomes align on the metaphase plate, which is an imaginary line that runs parallel to the chromosome poles. That is nothing but what happens in the metaphase plate. So much if you write also, it is enough. Next, moving further, differentiate between mitosis and meiosis. So mitosis, this is one of the important questions. Mitosis, it occurs in somatic or vegetative cells, whereas meiosis only takes place in the gametic or the reproductive cells. That is the male and the female gametes, they usually occur. Next, mitosis, it completes in one sequence of phase, that is in mitosis itself. But in meiosis, it completes in 2 sequence of phase, wherein meiosis 1 and meiosis 2 is there. Mitosis, it forms 2 daughter cells and they are diploid, that is, they are 2n in number, whereas meiosis will result in the formation of 4 haploid daughter cells. Next, mitosis, prophase is short and completed in 1 step, but in meiosis, prophase is long, wherein it involves 5 steps. 5 steps are involved in prophase of meiosis. Next, mitosis, crossing over does not take place, but in meiosis, crossing over occurs between the non-sister chromatids of the homologous chromosomes. Their crossing over occurs. So that is about meiosis. Next, moving further, what are homologous chromosomes? What happens to homologous chromosome during meiosis? So homologous chromosomes are nothing but pairs of chromosomes which are similar, having the same set of genes or same set of traits. That is how you can write. And during heterotypic division of meiosis, that is in leptotene, the chromosome threads that are there, they coil, and during zygotine, the homologous chromosomes that are there, they will start to pair. So what happens is, we are writing that is only to the homologous chromosomes. In packetine, what happens? The chromosome shows thickening and shortening. Diplotin, what happens? There is cessation of attraction force between two homologous chromosomes. Uncoiling of homologous chromosomes will occur, but they remain attached. That is, the non-sister chromatids of two homologous chromosomes that are there, they will form chiasma. So therefore, only in the chiasmata region they tend to remain attached. So that is what happens. So next, moving to anaphase 1. So in anaphase 1, the centromere of homologous chromosomes that are there, that is bivalents that were formed, they repel each other. And after separation of the centromere, these homologous chromosomes, they start to move apart. That is what happens in the anaphase 1. In telophase 1, what happens? The chromosome reach the opposite poles. That is, the homologous chromosomes reach the opposite poles and they become shortened. So what happened to the homologous chromosomes during meiosis is what we are writing. So I hope you understood. Let me explain once again. So what was the first question? What are homologous chromosomes? So what are homologous chromosomes? Homologous chromosomes are pairs of similar chromosomes having the same set of genes or similar traits. So much. That is called as homologous chromosomes. So this is the answer for the next question. So next question that is being asked is, what happens to homologous chromosome during meiosis? So during meiosis, we know about prophase 1, prophase 2, that is prophase 1, that is meiosis 1 and meiosis 2 occurs in different stages. Now in that, wherever homologous chromosomes are involved, that is what we need to mention here. Say for example, in leptotene. So in leptotene, homologous chromosomes we see. During zygotine also, something happens to the homologous chromosome. In packetine also it occurs. So in each of the stages, what happens is what we are writing. That is what happens to the homologous chromosome. That is what we have written here. So much you write, it will fetch you 3 marks. So this was about the session wherein we learnt about, that is, what all are the contents in the chapter, and also we learned some of the important 3-mark questions that will appear from this particular chapter. So I hope you found the session useful. So we'll meet again in the coming session, that is with the continuity of the revision class, wherein we'll deal with the MCQs. So see you in the next session. Thank you.